Hello, and welcome again to another version of HIV and Aging, the 2021 version. It's my pleasure today to be speaking to you about biological mechanisms related to inflammation, thrombosis, and COVID-19. This is my outline. We'll talk a little bit about introductory material, HIV infection as background, some other viral infections and thrombosis, and then get into SARS-CoV-2 infection and thrombosis, think about possible mechanisms and some conclusions. So viral infections viruses certainly kill normal cells in both powerful innate and adaptive immune responses. Many viruses can also break tolerance and induce self-auto-antibodies to self-antigens. Coxsackie viral-induced autoimmune myocarditis is a well-known example, and SARS-CoV-2 is no different. 15, now almost 20 different autoantibodies have been identified, including antiphospholipid antibodies and anti-interferon and other anti-cytokine antibodies. These will become important later on in the talk. A vaccination can also induce autoantibodies, and there's a very rare but clearly observed vaccine-induced immune thrombotic thrombocytopenia, where autoantibodies to platelet factor 4 heparin complexes occur. As for tolerance, many viral infections induce thrombosis, and SARS-CoV-2 very early on had a clear association between D-dimer levels as a measure of thrombin production, where 71% of non-survivors in Wuhan had a coagulopathy compared to less than 1% of survivors. This has been confirmed in many other studies, is better controlled now, but it's still a considerable issue. HIV infection and thrombosis is probably pretty well known to this audience. I'll cover it quickly. This is data from the Insight group showing the relationship of D-dimer levels, quartiles 1 through 4, and mortality over 96 months, showing almost a 10-fold increased risk in mortality in the higher D-dimer levels. So thrombin production is clearly associated, if not possibly causal, in some of these fatal events. And it's also true for venous thrombosis, where there's about a 1.4 increased risk, even after HAART came in the mid-90s, most of that risk remained. So these thrombin generation does seem to translate into clinical thrombotic issues. How about other viral infections? Well, here's some data, again, from the Insight group, after they pivoted and started looking at H1N1 infection. And what you can see is that in two different studies, the study Flu-002, which was the outcome was, it was an outpatient study, and the outcomes of interest were hospitalization, complications, or death from influenza, or Flu-003, which was an inpatient study, and the outcomes were worsening to the point where you went to the ICU or died. And these are third tertile versus first tertile odds ratios. You can see there's a strong inflammatory response associated with doing worse. That's IL-6 levels. There's a strong interferon response. That's IP-10 and interferon responsive protein levels, and a strong procoagulant response. There's D-dimer again, all being strongly associated in both studies with bad outcomes. There's other viral infections we could talk about, and associations with CMV, hepatitis C, associations with thrombosis, CMV, hepatitis C, and some others. Time restricts us, so let's get right into SARS-CoV-2. So what does the immune response in SARS-CoV-2 look like? Well, here's a sort of quote-unquote perfect example of a generic infection. The green is your innate immune response. Then this red line suggests the adaptive immune response from T cells, cellular and humoral antibodies, and it should all look like this, and your viral infection just sort of disappears rapidly. in the average SARS-CoV-2 infection, there is persistence of the viral load. You do get resolution of that innate inflammatory response, and you do get T cells and antibodies produced. But in some SARS-CoV-2 infections, those severe ones, we see persistence of viral load that's pretty unchecked. We see persistence, and this is the cytokine storm that people have talked about, of a pro-inflammatory response. We do see antibodies produced, but a surprisingly low T cell response in many cases. So there's something very different about these severe infections, and these are the ones that are most associated with thrombotic events as well, probably in part mediated by this prolonged and very significant unchecked pro-inflammatory response. And here are some data to talk about for both ICU in red and non-ICU patients. Interestingly, the pro-times and APTTs are not particularly noteworthy, but here's D-dimer days after admission on the x-axis, showing strong progressions in both cases. The non-ICU is missing from day 14 in all cases because they were often released before that time point, so the data does not exist. Here's a platelet count. And here's fibrinogen as a pro-inflammatory marker like CRP or others might be. So you're seeing that pro-inflammation going along with procoagulation and thrombin generation. The key points these authors make are that race and ethnicity seems to have major effects, not shown on this slide, but there are lower risks in Chinese compared to Caucasian individuals. Severe infection has a significant coagulopathy, which correlates with disease severity. And despite increased D-dimer levels, progression to over DIC in Caucasian patients on low molecular weight heparin is very rare. So there is this production. These are severe patients, but they're not getting DIC. And so we have to be looking at other potential sources. So some possible mechanisms here. Well, in HIV, Danny Dewick and others have well established that endotoxemia can occur as the Peyer's patches and the secondary lymphoid tissue around the gut is destroyed by the virus. You can see this in SIV infections in primates in just a couple of weeks. Peyer's patches are gone. And this helps participate in a loss of mucosal structure there and an increased endotoxin levels in HIV positive patients. This endotoxemia is procoagulant, and that is well understood as a mechanism. We've published another mechanism, including the modeling in a model that's been developed over the last 30 years by measuring procoagulant and anticoagulant factor levels and modeling them mathematically to show what likely thrombin production would happen. I'll just take you to this upper left-hand graph as an example. In untreated participants in the SMART study from the Insight group, you can see thrombin generation in the red bar is quite a bit higher than in the ART treated folks in the green line here below. And that's true if you then take the same people and compare them ART treated versus untreated, where we had the ability to do that. We believe this is this increased thrombin production with viral replication is actually mediated by interferon response. So the virus causes interferon response. There are many proteins that are changed, over 2,000 based on interferon production. There's a general reduction in protein synthesis, which is antiviral because the virus needs protein synthesis to reproduce itself. So this is a good thing. But that we have identified alters the coagulation factor balance, leading to a procoagulant state, which is a bad thing, giving us thrombosis risk. Probably well tolerated evolutionarily because this increased thrombin actually can help increase interferon production. So it's complex, but we believe this cycle can also play a role in thrombosis in viral replication. Also recently pointed out by Yogan Kanthi, Jason Knight, and colleagues that there's a lot of autoantibody production, as I mentioned earlier. Almost a quarter of COVID-19 patients are showing significant levels of antiphospholipid antibodies, which are procoagulant in many cases. Wolfram Ruff and colleagues have demonstrated that special ones, certain antiphospholipid antibodies, can bind to receptors on the surface of monocytes and dendritic cells. On monocytes, they cause the expression of tissue factor, which is procoagulant, and on dendritic cells, they get more interferon production, which leads to more antiphospholipid antibody production. So it's, again, an interesting cycle here that has procoagulant potential. So in starting to summarize mechanisms, we think there are two aspects to consider: the acute infection and the 25 to 33% of convalescent patients who get post-acute problems, the so-called PASC, or post-acute sequelae of COVID-19. We have to be thinking about both of these as we structure research studies moving forward. Microbial translocation that we see in HIV, could this be happening in COVID-19? Yeah, there are microbiome alterations in COVID-19, and some patients show persistent fecal shedding. So it's very potentially likely that we're getting some alteration in the gut that might allow for microbial translocation. The altered coagulome, well, as far as we can see, it's just the interferon response that's critical, and there's certainly a very strong interferon response in COVID-19. So this is another likely candidate for the thrombotic problems. And then maybe more specifically, antiphospholipid antibodies, anti-platelet factor 4 heparin antibodies. I didn't get a chance to show too much about this, but this has been a lot of discussion about vaccines, but certainly it occurs in natural infection as well with SARS-CoV-2. And then persistent endothelial, endopulmonary epithelial damage. That's a whole big area of discussion that we didn't have time to get into today. So some conclusions. Well, COVID-19 thrombosis remains a significant issue. Despite heparinization and the benefit that heparin gives from a clinical DIC and clinical thrombosis standpoint, thrombin generation appears to be somewhat unchecked by heparin, and there may be other sources that we have to be looking at for what that thrombosis is coming from and why it might be important. The full extent of its clinical impact, both in the acute and chronic phases of SARS-CoV-2 infection, remains to be established. Anticoagulation helps clinically, but does not control, as I just said, thrombin production in all cases. Its use chronically is under investigation in some NHLBI-funded clinical trials, and multiple thrombotic mechanisms are likely. Some are general viral infection mechanisms, others seem more specific to SARS-CoV-2. And I'll end there, and thank you for your attention.